Thank you very much, Freddie. And so Oxford's not completely unfamiliar to territory because I did my SHO years here in the days when we were doing 1 in 2s and 1 in 3s, and met my wife here on the cardiac wards. So it's definitely familiar territory. And I feel a bit overdressed, as I was saying. I don't normally wear a tie, and I'm only wearing waistcoats because I can't be bothered ironing my shirts. It's a good tip. Okay, so if you want to contact me about any of this later on, that's my email address. I'm very happy for you to do so. We're funded by a variety of sources, and most recently by NIHR as well, in fact. So I'm going to meander here through really just some questions about how you take a difficult treatment and get it into people. Something that's been worked up in the laboratories, but isn't a straightforward drug or straightforward device, but something else. And this really falls into this category that's labelled in Europe and Britain as an advanced therapeutic medicinal product. And there's a definition here. And I'll go through this a bit. So it's difficult enough getting drugs and devices into people, but when you're in this kind of rapidly advancing, emerging field where, people are making up the regulations and the ways of translating as they go along almost, it's even more challenging. So this is a path, and I haven't got all the answers, as you'll see. There are a lot of problems out there too. So ATMPs are gene therapy, somatic cell therapy, or tissue engineered products. And tissue engineering is where you're generally taking a scaffold and either implanting it alone or with cells in order to create a regenerative result. Now, not all ATMPs are regenerative medicine. So regenerative medicine, there's various definitions of it. This is a relatively simple one, where we're replacing engineering or regenerating human cells, tissues, organs to establish normal function. So this might extend, for example, from trying to create a new conduit, be it a bit of esophagus or a bit of blood vessel for a child who we know in utero hasn't got those things developed properly, right through to trying to replace whole organs and move on from where we are with conventional transplantation with all the problems that surround that as well. And the regenerative medicine toolbox contains a lot of things. It contains a lot of the things that are in the previous slide, in fact. But clearly not all gene therapies and cell therapies are regenerative medicine, but they are part of this toolbox. So you have cell therapies, gene therapies. We've got material science, where we have both biologic scaffolds, that is scaffolds which are based either on decellularized materials, which is where you take something from an animal or a human and strip out the cells so it's non-allergenic anymore. Or alternatively, you can generate such biologic scaffolds by taking cells and allowing them to make their own extracellular matrix, and then either leaving them in or moving them out. So various ways in which you can generate scaffolds. Then there's synthetic materials, and there's a MRC hub, which is looking at how we can actually generate new biomaterials which combine the best of biologics and the best of the controllability and printability of synthetic materials as well. So this is highly multidisciplinary. I mean, it involves everybody from engineers, chemists, cell biologists, gene therapists, clinicians, and surgeons. Highly multidisciplinary. So as surgeons, we deal with a wide range of problems that can be addressed by regenerative medicine approaches. And possibly the father of this, although I get most people in any brand new field tend to have, kind of starts and stops. This is Tony Otala. So he first started creating patches for bladders first of all in Boston, but then in Wake Forest, North Carolina, in the late 1990s. And had a successful trial of such patches in a small group of children in the early noughties. He's then tried to scale this up into adults by creating even bigger patches. What he does is he takes collagen, compressed collagen, and seeds onto it the child's own epithelial cells on the inside and muscle cells on the outside. And very early on, he created a company to help him do this. In fact, he's encountered a couple of problems which I think are symptomatic of some of the bigger issues I'm going to raise later on. Firstly, there's a problem of scale-up. And once you're dealing with very small patches, babies this big, bladders that big, patches that big, angiogenesis can occur fairly quickly, and infants of that size regenerate very easily. But once you're into adults, it's a different ballpark. We don't regenerate like children, and they're much bigger areas to cover. And then the whole issues of remodelling, scarring, immunity, and angiogenesis critically become very problematic. So that's one of the problems. And the other problem was this tension between trying to do something in a university and then working out how you could actually make it commercially viable later on. And that became a big problem for Tony's group, actually. However, he's still regarded as the father of surgical tissue engineering by many. So the valley of death is actually it's a phrase that's been coined in industry for where you've got a really good idea and you can't raise the money to get it into actually delivering it to people commercially. But it's been borrowed by the biomedical field for the phase between late preclinical work, where you've got something that works well in an animal model, preferably a large animal model, and where you actually get it into the very first human beings. And there's a lot of reasons why historically it has been a valley of death. Not least of these is that the science required to make that jump is not desperately interesting science, and it's not been particularly fundable in the past. So you have to do things over and over again in order to produce something which is fit to put in a human being. And this is called GMP, good medical practice. The process of making something into a GMP product is laborious, it's very expensive, it's very boring, and it's not interesting science. And so historically there haven't been the funds to do it. There's also not been an industry to pick up that gap either. If you create a drug, historically, there's been a very well described pathway where big pharma will swoop at fairly early stages. Now, there's a big drop-off, of course, so only 1 in 100,000 molecules that are mooted as drugs actually make it through into something that's actually used routinely in the clinic. And there's various steps at which it drops off exponentially along the way. But it's an established pathway. There's no big pharma in regenerative medicine. The big companies are starting to take an interest, but they're still watching the field. It's very early days. So you're left really with waiting for the environment, the funding environment, to catch up with the need to get people across in universities. And 90 to 95% of all translation regenerative medicine research has no commercial input and is done in universities still today. So rather than the valley of death, however, I view it more as a steeplechase, where we know what some of the hurdles are, and we know we're going to get to the end. We may trip up from time to time and certainly get wet, but you know you're going to get to the end, even if you don't win. This is a conventional model of the translational pathway. I borrowed this from John Tuck, who was our vice provost at UCL. And within this, you may have seen, there are lots of variations on this, from discovery science down at one end on your left, down to population health and, actually really improving large-scale population health at the other end. And then they've got this blue arrow which connects the population health back to discovery science. And actually, technically, you should probably have that blue arrow at every one of the other boxes as well, because there's no reason why you shouldn't be feeding back into discovery science the whole time if you've got human material to do so. And that's a whole interesting area itself called reverse translation. So even if we don't progress at any of these stages, it's beholden on us not only to examine the results of the work that we've done, be it in the large animals, the small animals, the first-in-man studies, the phase 1-2 trials, phase 3, use that material and ask questions about exactly what's happening mechanistically, as well as what's happening to the patients themselves, and feed it back. Within this pathway, there's said to be 2 big translational gaps. And the first one is the one I've described, the valley of death, between the bench and the bedside. The second is between actually proving that something works according to randomized trials, or in the area of rare diseases, if you've got a very large-scale post-marketing surveillance and big series. and it actually being properly implemented. And I'm sure you'll be aware that it takes something like 10 to 20 years from proving that a drug is truly effective in an area, even something as common as cardiac disease, to it actually really being used everywhere in the way that it should be. So 2 big translational areas there. So you've got a really good idea in the lab, or you're working with a scientist who says, I've got this fantastic thing. So this, whatever it is, pancreas in a box, for example. How am I going to get that into human beings? So what you should do, once you've got a really, really good idea that has proof of principle in small animals, is to start thinking about the translational pathway as early as possible. And in particular, start talking to a wide multidisciplinary group of potential collaborators and funding bodies, but also regulators. In America, they have several stages of talking to the FDA, including IND, investigational application, back to pre-IND, back to pre-IND, and they've even got pre-IND, which is where you simply have a very, very rough chat. And talking to regulators at a very early stage will help prevent some of the problems that you might get if you don't do so. So, for example, designing your animal model in a way that conforms to the 3Rs principles, but at the same time will deliver the sort of information required from the clinical trials application is absolutely critical. And you need to do this in an appropriate animal model, but also to what's called GLP standards as well, which is more expensive than doing it without GLP, but is well worth it, and does need thinking about. So this guy is viewed as the father of mesenchymal stem cells, and they're the stem cells that are mobilised from all parts of the body, but particularly bone marrow, in the event of trauma or inflammation, and they home and support repair. They're the ones that we use routinely in our own stem cell-based work. And he said the FDA is inhibitory to translational stem cell therapies. There's no sense in regulating them as a drug. Well, whether we like it or not, at the moment they are regulated as drugs. But they're regulated as a special class of drugs, these advanced therapeutic medicinal products. And the FDA are still grappling with exactly what that is, but actually the EU have very good definitions. I've actually found along this pathway of translating these things that one of the lowest hurdles has actually been the regulators, particularly in this country. The MHRA are hugely overstretched, but they are extremely willing to learn and engage. And they're very open to discussions. So if you are working in drugs or cells or genes, do go and talk to them, because they are extremely helpful. And the turnaround time for MHRA applications has come down enormously now. So for something where you really have discussed it and you've got a really nailed down clinical trial as application, you can have a turnaround as short as 3 months sometimes. They, of course, are having to grow at the same time as the rest of the field. So as the field is developing, they're having to put in new expertise as well. And that kind of dialogue is doubly important because they're learning at the same time that we're learning. So I actually have nothing but praise for our regulators, in fact. They're not the kind of hurdle that I think the FDA still remains in America. So this guy is actually the person who really makes everything happen. This is a guy called Mark Liddell, and he is what's called a QP. Now, there aren't many QPs in the UK, and that is a problem, particularly people with his kind of experience. A QP is a qualified person. There's somebody who has a license to supervise the delivery of advanced therapeutic medicinal products and runs an appropriate GMP facility. So the GMP process has a pre-GMP phase where you're getting everything ready and making sure that all your processes are as repeatable as they possibly can. That itself is a problem, by the way. If you've got a drug, then you would expect the consistency of production of the molecules to be correct with every batch down to about 0.001%. So you'd expect to be getting exactly the same sort of propranolol every single time. The same cannot be true of cells, particularly if they're autologous cells and they're coming from yourself, because we're all so different. As we age, our cells don't multiply and grow as well, and that's a big issue. You could get around that by having panels of allogeneic cells. And in some contexts, of course, that works because some of the areas of the body are privileged sites immunologically. So for the eye and the brain, and to a certain extent joints, allogeneic cells can work pretty well. In any case, the cells you put in probably only hang around for 6 to 8 weeks, in any case, having done their job. So that pre-GMP phase is absolutely critical in working up. GMP labs themselves are a big issue. We don't have enough capacity in this country to deliver all of the cell, gene and tissue therapy trials that we need to. And we do need more capacity. But the wrong thing to do is to set up lots of little ones. And unfortunately, that's what tends to happen, because they're very expensive to build and staff, and therefore the temptation is to have one for haematology, 2 or 3 clean rooms, another one for medical devices, 2 or 3 clean rooms, one for orthopedics, one for eyes. It's incredibly inefficient. If you have one room, as they have at Queen's Square, the neurological hospital in London, it costs you about $500,000 a year just to run the facility before you staff it, for one clean room. And then you've got to have downtime, and you've got to refurbish it every few years to meet modern standards. And occasionally you get infections and you can't use it. So one room is no use at all. 2 rooms is little better. 4 rooms you could just about get a by with, and you could run trials of 4, 6 people maybe, something like that. But if you really want to do proper phase 1, 2 clinical trials, you need to have critical mass. And that's still an issue that this country is grappling with, I think. We happen to have that at the Royal Free, where we have about 14 rooms, which are categorised for all preparation. And the products from there actually can be produced for clinical trials all over the country and indeed internationally. So some are shipped out to Europe and so on. So it's an expensive business. Like I say, it's not interesting science, but it's absolutely critical to get something that is as repeatable as possible, as high quality as possible, and is produced in a facility that really know what they're doing. Having got to that point, you still need to engage with where you're going to take this later on. And part of the workup to clinical trials should always be a feasibility phase. So you'll have heard this before, I'm sure, but you need to be thinking about who the target population are and engaging with them themselves. So actually getting patients involved, even at this early stage, is very valuable. It's not only something that the funders like, so actually having patients in there in the design, NIHR require it, but other funders like to see it there too. But it really does help you focus your mind on exactly not only what the diseases that you want to treat, but what really matters for patients. And what matters for patients is not always what you think it is. So, for example, with our airway trials, we discovered quite early on that the most overwhelming problem that people had was fatigue. We thought it would be the breathing or the voice or the swallowing, but actually it's the overwhelming fatigue that they have. So then you can target your outcome measures specifically and create new outcome measures if necessary, and they can help you design the patient information sheets and make sure everything is how it should be. But feasibility work doesn't end there. You also need to engage with the clinical colleagues who might be applying it too. Is this something that they feel they would use? How would they use it? Would they engage in clinical trials if it was rolled out to them? And how much would they and their institutions be prepared to pay for something like this? Because these things are not cheap. You might also want to start down the commercialisation feasibility pathway too, and start thinking about how can you commercialise this. Now, in rare diseases like the ones that we've been dealing with, it's difficult to see how you could ever make a big profit from that. But there are ways in which you can embed it within a national health service such that it does actually pay for itself. There are models, both commercial and embedded in the NHS, that can work for these things. And finding commercial partners is very difficult, as I say, because there simply aren't that many of them. But if you can, they will have the business heads to help think about the future and where this might go. And particularly kicking on from those early phase 1, 2, the safety and potential efficacy trials into the bigger trials later on. So IP, this is really difficult. And I think this is actually starting to hold back some of the things that we can do in the UK and internationally at the moment. There's a big obsession around IP in all universities. And they're vamping up their business offices as far as they can to try and eke out every single bit of IP they possibly can from the basic translational and clinical scientists that they're working with. But in an area like this, where you're actually dealing with combinations of things and a lot of things that are already out there in the public domain, it's difficult often to define where the IP actually is, because so much of it's been already published. And therefore, it's more important that if you want to create money out of these things, that you're first to market. So once you're actually there, the IP is not that important. It's actually very difficult for the second wave of people with something else who may have IP to actually get into the market that you're already in. So there are other issues around that. But collaboration between the universities in the UK is becoming increasingly difficult because of this, and I do think it's a big issue. I know in Bristol, they're actually suddenly, they've clamped down on this so much that it's almost paralysed their ability to roll out the grants out the door. So just a little bit on the work we've been doing. I became very interested in airways, as you know, and so it appeared on the surface that building a trachea would be a logical step up from where Tony Otala was. So he was creating flat surfaces. If we can just create a simple tube, it would be really quite straightforward. And there are patients who have need. It's not a big group of people. These are small numbers, but they are really disabled and with big difficulties. They're dependent on metal tubes to hold open their airways or tracheostomies. They can't get out there. They're just paralysed by fatigue. They're often young or middle-aged. And they have a reduced lifespan because stents erode and they get pneumonias, and it's generally very distressing. So there is a population out there, and we don't have an effective replacement for airways. Unfortunately, the trachea is not just a simple tube, principally because it's exposed to the air. And therefore, if you put something in, you've not only got to deal with the biomechanical issues, but you've got to deal with the fact that it's got this interface which is potentially and actually infected. So we've not only had to work very hard on the biomechanics, but also the process of epithelialization as well. And we've done this in a stepwise manner. We started, as Freddie said, by doing a number of basically case reports. So we did an adult in 2008, we did 2 children, one in 2010, one in 2011. And we did these on what's called compassionate use basis. So most countries have compassionate use law. And this is where, if you've got good a priori evidence that a new technology might work, and you have somebody who is in dire need and usually is going to die for lack of the functionality of conventional methods, that you could apply this new technology under certain ethical boundaries to these people with fully applied consent. So the rules are that you have to have dedicated consent and you have to have it externally monitored. You have to go through the hospital's clinical ethics committee as well, and you have to notify the MHRA, although they're not actually involved in the regulation of compassionate use. And that's the overall framework, but it's not standardised across the UK, and it's certainly not standardised internationally, which has created great confusion about some of the things that have happened in other countries, like Germany, Japan, and Sweden. We're pretty good at it here, and this kid was done at Great Ormond Street. He had a windpipe that was congenitally narrow. He was kept alive until the age of 10 by a metal stent that eroded into his aorta, and he woke up one morning, coughed, and blood started pouring out. My colleague Martin Elliott's a very skilled cardiothoracic surgeon, was able to repair the aorta, but we had to replace his whole trachea. And there's no conventional technology that can do that. And so we applied a tissue-engineered trachea to him, and he's still alive and well today, and growing and doing well. So compassionate use can be very useful, but it can also be abused as well. And there are lots of questions about how it's done. Also, these patients are in extremis, they're rare, they're one-offs. They're not research patients per se. You're not allowed to then continue to follow them and biopsy them to get more information. What are really needed, of course, are phase 1-2 clinical trials, actual proper clinical trials. And getting from that point to the clinical trials has taken us about 7 years. So what we did was we went back to the lab. We've done an awful lot of work, as I say, working around biomechanics, means of decellularization, and also epithelialization, producing a number of products and combined products to support airways. We've been particularly interested in the process of angiogenesis, which is complex and occurs in phases of maturity. And we've now got some very good animal models for studying how this happens. And we think we're able to enhance the quality of the vascularity and not just its quantity. In practice with the trachea, because it's so thin-walled, it picks up a blood supply within 6 weeks and it optimises within 2 months. So it doesn't tend to be a massive issue, but once you get into bigger organs, thicker organs like larynx or kidney or whatever, it's then massive. So we moved up from our small animals into our big animals, and this is a pig larynx where we've replaced a third of the larynx, including one entire vocal cord, false vocal cord and subglottis. And here the pink is remodelled cartilage, and the white is bone. The mesenchymal stem cells that we put in, you can control what they turn into to a certain extent, but what MSCs and other progenitor cells decide to do in vivo is not just dependent on what we want them to do and what we try to make them do in vitro before we put them into an area. It's dependent on the biomechanics of the area that they're in, and also the chemical cues that they're getting from the other cells and the extracellular matrix around them. And that's why some parts of this are forming cartilage and some are forming bone. Another example would be a group in France where they've actually used aortic scaffolds to rebuild bits of trachea, and have found that over time they're actually getting cartilage forming within an aortic scaffold, which kind of doesn't make sense, because why would you get cartilage in aorta? It only makes sense when you consider that the local progenitor cells are actually reacting to where they are in space and the mechanical and chemical cues that they're getting. So on the basis of all of that work, we've got 2 clinical trials at the moment, 3 clinical trials actually. This is Regenvox, where these are partial laryngeal replacements, and we've got our first implant coming up in about 5 weeks' time. Assuming nothing gets in the way in the meantime. And there's Inspire, where we're going to do 4 tracheal implants. Now these are phase 1-2 trials, so clearly there needs to be a change in terminology. You can't do a phase 1 trial in the sense of a pharmaceutical product with these kind of things, or indeed with any surgical implants, because you can't put them into volunteers. So it does need a different type of terminology. But the phase 1 bit still refers to safety. 2A refers to a non-randomised small-scale trial in actual patients. So that's what these are. And we're not going to be planting 15 patients, we're actually going to treat 4 to start with. 15 is the number that we're going to need to treat to receive an EU license to be able to apply this generally as a licensed medicinal product, either within the health service or within commercial sector. So we only need 4. When you're doing something completely new for the first time, especially surgery, it's incremental. And so an important part of this, and the reason why we've chosen 4, is that we know for absolutely certain that every time we do this within those 4 patients, we're going to be changing something. We'll have learned from the first one when we do the second one, we'll learn from the second one when we do the third, and then we'll learn from the third when we do the 4th. And the idea is that by the time you reach the 4th patient, you're actually in a position where you have not just all of the extensive work that I've shown you before, preparing a GMP product with quality control and release criteria, and making sure that every time we produce a scaffold, it's very much the same as the last time we did it and all that. All that's nailed down. But we've also nailed down a surgical process as well. And when you actually then go on into your phase 2B and the post, the surveillance, the follow-up studies, you've then got something that's as standardised as it can be within surgery. And we have funding to do that should we be successful with our 4 patients. We do have funding from Horizon 2020 to roll this out across 4 countries in Europe as well. We're not the only people engaged in regenerative medicine research. There are lots of other groups around the world as well, of course, working in all kinds of areas. So groups in the States have produced a heart like this. This has rat stem cells in it, which have created myocytes. And this has a tiny cardiac output. It's actually really, really difficult to scale this up into a big heart. To decellularize a whole pig heart, for example, takes about 3.5 months, and repopulating it takes another 3.5 months, and even then you can't really get much of an output. But what they have been able to do is create cardiac patches for people who've had infarcts, and there are trials of these going on. People have also been able to make lungs that have ventilated rats for a couple of days, kidneys that have produced urine for a couple of days, and livers as well, which have sort of function like livers on small scales. But as with Tony Otala, scaling this up, still big challenges for these. People get very excited also about bioprinting. So wouldn't it be wonderful if we could just get the cells that we want, all the different types of cells, the extracellular matrix, and maybe some biomaterial, and just print all of that out into a kidney? And actually, there's a TED talk where Tony Otala does that on stage, but obviously not using this. He uses one of the standard bioprinting materials. But his group have actually produced this thing called ITOP, which is able to produce to print two sorts of cells and two sorts of different materials at the same time to create a new construct. Again, very small scale. Here they've produced a bit of calvarium to replace an area in a rat's skull. They've also created a small muscle on the back of a mouse. And as everybody does, they made an ear as well. They put on the back of a mouse because that's what people do in tissue engineering. Okay, good. So innovation in surgery is difficult. And we're kind of in a kind of transition phase where we're really analysing the way that we do everything. Here are two surgical innovators, if you like. Who's this guy at the top? Yeah, it's Christian Barnard. And do you know who this guy is? That's Macchiarini. Okay. So we know about Christian Barnard. So he was a cardiothoracic surgeon from South Africa who went and spent some time in Boston. While he was there, they'd spent 20 years working up towards that heart transplant. They hadn't quite got to the point where they were going to do their transplant trials. And he looked at what they were doing and he said, well, actually, all this regulatory faff is a bit not terribly important to me. I'll go back to South Africa and just do it. And that's what he did. So he went back to South Africa and incrementally developed heart transplantation. And clearly this has made a big impact. He was the first person to do it. He did develop the technology and the methods incrementally. It would be very, very difficult to do that in this country today in that way. In particular, you do need to have very tight consent processes and ethical control around what you're doing. Unfortunately, this guy, he felt that he was in the same category in the modern world, and he's not. So he went to Sweden, where he started putting in tracheas made of a synthetic material, which had no preclinical evidence for it whatsoever, without any ethics, without proper informed consent, in patients who really weren't suitable for it. It's astonishing that actually happened. I mean, just saying it kind of makes my blood run cold. And there's a documentary on him. But it did actually happen. And it's highlighted a number of areas where we really can't afford to go in future. This is a paper by somebody called Schwartz, who's a paediatric surgeon from America. And she has outlined a number of ways in which we can move surgical innovation, even complex, high-risk things, into people in ways which are measured and ethical. And she describes three areas, really. The first is where we're making small incremental changes to established procedures. So, for example, every time I do a neck operation, moving nerves around for laryngeal paralysis, I might think, well, actually, I've been doing it this way for years, but actually, if I move that nerve to the left, actually, that's going to make my life a lot easier. And then I could publish that as a case report. Now, that kind of improvement is enshrined within the normal consent process. Our patients do allow us to make minor modifications to surgical procedures as we're going along. Now, where that minor procedure becomes a major procedure is less well defined. But that incremental change actually is fundamental to progress in surgery and ought to continue. At the other end, there are the actual clinical trials, like the ones that I've been describing, where you've gone through all the regulatory processes and you've got your ethics and your R&D and the funding. These are very expensive, very laborious, a lot of paperwork, but they're the gold standard. And it's what we should be aiming towards for most things. But in between, there is a grey area. And this includes things where, like orphan diseases, where there aren't that many patients in a particular area, and where there's no conventional treatment that's available for these people anyway that's effective. And in that grey area, we still need to have regulation. And the compassionate use regulation is a good start, but it's not tight enough. It does need tightening up. This is the framework that she proposes for such a grey area. That if you're going to do things like that, you first of all need expertise to do it. You need technical skills, obviously. You need to be aware of what the potential hazards might be, all the potential hazards, and make the patients aware of it in a proper process of informed consent. There should be no conflict of interest. And I don't know, there are some obvious conflicts of interest that you need to avoid, like, making money out of every time you do it. But actually, we all would want it to work. And that kind of conflict of interest is slightly different. We need to analyse what we do. So it's critical that if you're going to do something new, that you measure everything you possibly can about it. And, importantly, you publish it, whether it works or not. In a sense, it's more important to publish things that don't work. It's well known that in the area of pharmaceutics, historically, 90% of drug trials that show a positive effect of a drug get into the literature. And 90% of those that show a negative effect of a drug don't make it into the literature, which is outrageous. We need to know both sides of things. This is Peter McCulloch's development, the IDEAL collaboration, who are also thinking about ways to engage surgeons in this gradual improvement process. And I'm hoping to work more with Peter on this as we go forward. So the next important step is working with the best possible clinical trials unit. And happily, you have the best possible clinical trial unit here, without a shadow of a doubt. And having people who really know what they're doing. And thinking about how you design these early phase clinical trials. And that too is an evolving area. The gene therapists are actually leading the field here in design. So where you've got a rare disease with a small number of children with a genetic disorder, it's going to be very difficult to do proper power analysis. But you can still do dose escalation studies. There's still ways of doing that, even in very small numbers of patients, where you actually measure the effect at a certain level of gene therapy and then gradually work your way up. So it is possible, but we do need to continue to work with trialists, professional trialists and statisticians to think of new ways of getting the most out of limited numbers of patients in these phase 1-2 clinical trials. We also, of course, need to work with our hospitals. And happily here you have a biomedical research centre which is not only functional, but actually does what it says on the packet. Most hospitals don't have biomedical research centres or have biomedical research centres that may not be quite so effective. So to do a clinical trial, again, very early on, you need to start to think about every step of the patient pathway, every single step, from what nurses are going to see them in the outpatients to what radiology they're going to have. Are they going to have physiotherapy? Are they going to have speech and language therapy? And you need to engage with all of those people. And that's very time-consuming. These are all very busy people. And most of the time, they don't have research built into their jobs. So you might have to consider building that into the grant if you can. So you can see the grants are starting to stack up in cost here quite a lot. So list all of the stakeholders, wherever they may be, and get them involved. I used to rely on research and development offices to give me the lists of prices and to notify these departments. But they are pretty snowed under. They're getting much better, but they are not sufficiently sophisticated to do that. You still have to engage each individual department yourself on that kind of level and make sure that they do have the capacity. So, for example, in one of our trials, we found that a hospital had signed off on doing a clinical trial, but actually nobody had told the speech and language therapist. And there wasn't enough CT scan time. So actually, although it had all been signed off by R&D, these boxes hadn't been ticked. Things hadn't filtered down. So that process needs to be streamlined. R&D is getting streamlined. There are time limits on MHRA approvals, and there are time limits on ethics approvals now, and they're really very good and very efficient. But historically, there have not been time limits on R&D. That's recently changed in the last 6 months. So R&D now have the same 60-day turnaround time as everybody else, and it's about time, because previously things have languished in R&D for absolutely ever. But our hospitals are still very busy. And, you know, there's it's 700 million deficit in NHS? It must be more than that. I think 700 million is probably one hospital's deficit, actually. It's an enormous amount. And we're snowed under with patients. So UCH, where I work, is completely full of cancer and chest infections. And it's virtually impossible to get any of my routine patients in, let alone a clinical trial patient. And so, again, we have to think about ways in which we can do innovative surgery in an environment that not only has all of the structures I've been talking about, but perhaps has also dedicated access to operating theatres and clinical space, which allows you to do innovative surgery, free from some of these constraints, and where research actually is a really high priority for the people in that area, that it's in the culture as well. So patient recruitment I've talked about already. So having patient and public involvement is absolutely critical, and helping them design everything. And you need to plan for and prepare for worst case scenarios as well. There's no point in glossing over that. And we do now. So in the light of Macchiarini, in fact, people became very concerned about what might happen to our tracheal patients. And although we've been through absolutely all of the stages of approval, so first of all, you have to get funding. So there are enormous panels of international reviewers and national reviewers who have to say, yes, this is worth doing, and the science is sound. You then have to get to MHRA, who say the science is not only sound, but it's sound enough for us to give you a clinical trials application. And ethics, who say, yes, actually, you're ticking all the ethical boxes. We're happy with the information you're giving to patients. And the R&D. So those are the conventional things. But people still weren't happy. Because I can see where they're coming from. If you've got a patient who's desperately ill in front of you, they can probably be persuaded to do almost anything, as Macchiarini unfortunately did. And so perhaps we need to think about a different way of doing things for the most high-risk trials. And what we've done is institute an extra level of patient protection. We now have an independent clinical MDT. Made up in our case, it would depend on the trial, but made up in our case of a speech and language therapist, an anaesthetist, a respiratory physician, and an ENT surgeon, who are unconnected to the clinical trial itself. So the patients see these people individually. It's very time-consuming, but they see them individually. And then those experts then come together. Oh, sorry, I missed a step. They have to be trained. So we have a training program for these experts. Where they come in and we teach them all about the trial, and they have opportunity to ask questions. And so they're not going into completely de novo. They have a formal training in it as well. They see the patients. Then they come together as an independent MDT, in which the patient's invited to be present themselves as well. The dynamics of a meeting where you've actually got a patient present is completely different from one where you don't. And I know increasingly, for example, and certainly in head and neck cancer, patients are being invited along to their own discussions. And it really does. It gets rid of all of the, the stroppy kind of big classic surgeon in the middle who's kind of being rude to everybody around him or her. And saying, oh yeah, he's got to have surgery. We've got to have surgery. And you radiologists don't know what you're talking about. If the patient's there, that kind of doesn't happen anymore. So the patient comes along to this MDT as well. And at that meeting, they discuss whether the patient, A, really does conform to all the criteria, and B, that the patient really does understand all the risks and benefits. And only if the patient passes that stage are they then formally entered into the clinical trial. It's, from the patient point of view, you can imagine all the steps they've had to go through here in terms of screening and consent, and then also this IMDT. There is a big risk of patient fatigue in this. But they're relatively small numbers of patients, and they are high-risk trials. And certainly our early experience is that it actually works. So you still get other problems, though. We're actually relying on the NHS Blood and Transplant Service to provide us with our scaffolds, which we then decellularise and so on. And one of the unexpected things, okay, maybe we should have expected it, was the difficulty in getting rid of infection. So what happens is a patient dies somewhere in the north of England, and they're a transplant donor, and when they've been through the normal transplant process, their body is transported to Speke in Liverpool, where there is an operating theatre for tissue retrieval, where they retrieve corneas and bones, which they then use for bone grafts, skin, but they're now retrieving airways for us as well. But that, getting the patient from where it's originally retrieved to Speke can take anything up to 2 days. And you can imagine how much infection is going to set in that time. So getting rid of the biofilms has been a really, really big problem. And although we were all set to go last year, it's taken us another 8 months to work out how to deal with biofilm on these things and get rid of them. But we've now done it. We now know what to do. There are also some interesting things that can happen to you that are completely unexpected. This is one of the unexpected things that happened to me, in that I'm kind of flattered in a way, but I've got an internet blogger who's pursuing me, and it's very entertaining. So this guy appears to be some sort of postdoc who didn't really progress, but it now spends his entire career attacking other people. And it is actually quite entertaining. So he called me the biggest liar in medical history at one point, which I kind of view as a badge of honour, but I think there are a lot of liars out there who'd be really disappointed that they're being missed out. Gregor Mendel, for example. Transparency. This actually does bring me to the next point. So you'll recall from the ethical criteria that Schwartz was proposing how important it is to get everything out there. And I think this is going to be the biggest change that's going to happen in, there's plenty of young people here, in your lifetimes in terms of medical publishing. And this is F1000. There are actually other fora that do this. So the conventional paradigm is that you write your paper, you then work out what is the highest impact factor journal that might, if you're very lucky, take it. You send it there, it's peer-reviewed, they bounce it. You then drop down your expectations a little bit. They bounce it. You drop your expectations a bit. They finally accept it with revisions, which take you a while to do. On average, that whole process, from your submitting the first time around to actually being accepted, takes well over a year. And a lot of things get lost in the meantime. There are other aspects to that too. So you don't know who the reviewers are. You can work it out sometimes, but you don't always know who they are. And nobody knows what the reviewers have said about your paper either. And because you've gone through different iterations, nobody knows what your original thing looked like. Most of the time, you're not actually publishing the original data either. My time's up? Okay, Karen, yeah, good. So that was the end. So, not always. I mean, there are a lot of publishers now will take all of your raw data and put it up there on an online supplement now. But that isn't always the case. This is a different paradigm altogether. So here what happens is you have your paper, you've done your science, you've written your paper, and you submit it to this forum or one of the alternatives. And this is the fastest growing area of medical publishing in the world right now. They then do a light touch screen. Light touch in the sense that it's got to look like a good paper in the way that it's structured. It's got to be written properly. There's got to be no major ethical issues that are obvious. And assuming it passes the light touch screen, which is done by a large team of professionals who do this all the time, it's then put immediately online and sent for referees. And the referees, they are also put online as well. So what they've said about the paper is then published below the paper itself, along with who the referee was. So there's immediate transparency, not only about the paper itself, but also the refereeing process. Importantly, all of the original data goes up there as well. So anybody who wants to reanalyze it to check whether they actually believe the statistics, or anybody who wants to do a meta-analysis in the future, has it all there straight away. It reduces that gap from acceptance, actually to publication is about 18 months in fact. It reduces the whole gap from your work being done to people seeing it by 18 months. Which is a massive amount of time, particularly in the career of a postdoc who's trying to get on, or in the career of somebody who's trying to get a big grant out there. It's a massive amount of time. But you can immediately see some of the issues around this. The big publishing houses have their big journals, and they have impact factors. And the metrics that drive the international rankings for universities and the metrics that are increasingly going onto people's CVs for appointments to senior positions include things like impact factor, H-index, citation indices. So there's still a drive to publish in those four instead. There is an alternative, though. So F1000, the 1000 refers to 1,000 scientific and medical experts, all of whom are right at the top of their game. And they are allocated papers as they come along to screen. And the most outstanding ones get given a star rating. It's a bit like Expedia or whatever. They get given star ratings. And this itself may in the future start to become a substitute for the impact factors and the H indices. But importantly, it is transparent, it's immediate, it's open, and everything's out there, including the negative papers. So, approaching the hurdles in this exercise, just a few key points. Please, please, please assemble the best possible multidisciplinary team. You're in Oxford. You're going to have some of the best people in the world in every single field. So look around you. But if they're not in Oxford, don't be afraid to go to wherever they are and build the best possible internationally renowned team, because that gives you the best chance of success early on. You need to have institutional support, and you know, by that I mean you need to have, you know, support of your department, university, hospital, to get things through those hurdles I've talked about. And you need communication with all of the stakeholders as well. So especially funders, they're very happy to talk to you, the regulators, the patients, the clinicians, and if appropriate with industry too. So tissue engineering today, actually all I think so. Oh, they've all, okay, so let's go back. Okay, that's fine. Fine. So that's what tissue engineering is today. It's a timeline, you can imagine. That's what's going to happen. I've actually got my animations wrong here. If we look at gene therapy, so I'm not a spring chicken. When I applied to the other place that I won't mention, it's a Fen-like organisation, there was an entrance exam, and the question I had to answer was, what is the future of gene therapy? And that was 1978. In 1972, gene therapy was first conceptualised, so people thought that it might actually work. And then people went back to the laboratory to see if they could actually get it to work in animals. Around 1980, the first gene therapy patient, so the first gene therapy was applied to a patient for the first time. And this was well reported in the journals and the newspapers. But there was no formal scientific report on it, and there was no formal follow-up on the patient. And in retrospect, people have asked a lot of questions about whether it actually happened at all. So that seemed to be a milestone, but people aren't quite sure exactly what happened there. They went back and did more animal research, and it wasn't until 1990, so that's like 18 years later, that there was a temporary success in a patient with a hematogenous condition, where there was some transient expression of a gene which allowed an immunodeficiency disorder to be managed more successfully for a few months. In 1993, they were able to make the first permanent genetic change in one of... there were two girls in America who were implanted, again, for a rare immunodeficiency disorder. So two patients, so a phase 1-2 trial, if you like, with two people in it. So you've got one patient about which there's a lot of questions, one temporary success, very similar to our 2008 lady in Spain, in fact, and then a couple of permanent changes, similar possibly to our child experiences. But at this point, there was then another further gap because of the reasons I've been talking about. So going from that point there, with a few index cases, which were successes, to actually rolling it out into proper clinical trials was very hard work. And unfortunately, during this period, because the clinical trials didn't immediately follow on, for very good reasons, and the ones I've been pointing out, it was branded a failure. And people said, gene therapy's not going anywhere. You know, what's the point? And funding bodies stopped funding it. However, in 2002, the first successful phase 1-2 trials actually did take place in small numbers. Again, hematological conditions. And that kick-started the whole field. So more and more clinical trials were done, largely in kids, largely in hematological disorders. And the first US-EU license, Glybera, was in 2012. Actually, the first licensed uses were actually in China, in fact, in about 2006, 2007. But this was the first product to be actually licensed by both the EMA in Europe and the FDA for use in 2012. So you can see this timeline here, 1972 to 2012. So when it's first conceptualized, my entrance exam, all the way through. It's a long, long way. And there are now about $600 million invested in the US alone in gene therapy every year. And there are more than 12 clinically approved products already in gene therapy. So getting to that point, for tissue engineering, we ain't there yet. So Atala started, has had some big issues. We've had a lot of hurdles to overcome as well. And there are others, getting hearts scaled up, getting kidneys scaled up and all the rest of it. But we will get there. It's got a long way to go. However, listening to people like Atala, like me, like Harold Ott, who's doing the heart work, like Doris Taylor, Steve Badilak, people who are out there trying to do these clinical trials and gradually overcoming hurdles as they appear, I think we can bring this down a lot, particularly if we work with our regulators and our funders and our organisations to do it. So it's not a valley of death. It is a race. We will get to the end. Of course, not all drugs that go through phase 1-2 clinical trials are proven effective. But actually, even if you lose this race, you still win, because you've still learned a hell of a lot about the science of getting something from late preclinical into human beings and what works and what doesn't. And others will learn from the failures just as much as they'll learn from the successes. Thank you.